research. My name's Dave Obrochta. I'm a member of the Gene Convene Global Collaborative team. And I'll be hosting and moderating this series of webinars focused on some of the technical aspects of the research and development of engineered gene drives, along with my colleague, Hector Kumada. Before we get to today's speaker, I'd like to say a few words to introduce you to the Gene Convene Global Collaborative. I want to say a few words about this and upcoming webinar series, and then I just want to say a couple words about how we're going to conduct today's seminar. The Gene Convene Global Collaborative is an initiative within the Foundation for the National Institutes of Health, and it's arisen from and really is an extension of the FNIH's long-time involvement in the vector-borne disease space. Gene Convene is working to advance the safe, ethical, and rigorous exploration of gene drive and other genetic biocontrol technologies by anticipating emerging issues and building consensus for best practices and guidance. Gene Convene offers accurate information, advice, technical support for the purposes of fostering reliable approaches to research and governance of gene drive and other genetic biocontrol technologies for public health. The potential of genetic biocontrol and gene drive technologies specifically to contribute to malaria's control and elimination, as well as other urgent public health challenges, is substantial. Gene Convene thinks that the responsible exploration of these technologies is critical in order to see if, in fact, these technologies can fulfill their potential. So I encourage you to learn a little bit more about Gene Convene Global Collaborative by visiting our website. It's fnih.org/Gene Convene. And before I move on, I'd just like to point you to the Gene Convene Virtual Institute. This is a knowledge resource that aggregates, tracks, organizes, and shares the latest about gene drive and genetic biocontrol technologies, from scholarship and research to media reports to policy and regulatory documents. This is part of an effort to create a resource where those interested in gene drive and genetic biocontrol can come and see what's happening and what has been happening. The Virtual Institute publishes a weekly newsletter that can help you stay abreast of the activity in the gene drive space. The gene drive space is actually becoming increasingly active. This year alone, there's been over 100 scholarly publications and many more media reports. One of the reasons the Virtual Institute was created was to help you keep up with all of that activity. So there are really important discussions and conversations that are ongoing concerning these technologies and their potential applications and their safety. The Virtual Institute can assist those with an interest in staying abreast of these important issues to do just that. So I encourage you also to follow us on Facebook and Twitter. Let me say something about this webinar series. The following this webinar, this webinar series is will explore the technical aspects, more focused on the science, and is intended really to provide the researchers an opportunity to share their latest results and to receive feedback and discussion from others. While this series is limited to 6 researchers in an ever-growing field of research, we'll be coming back to this technical theme in later webinar series. And we'll have an opportunity to hear from others that are also doing important and creative work in this space. The next webinar series that will explore the themes of regulation and policy. And we'll provide you with more details and registration information shortly as they become available. After the new year, we'll continue with other themes, and you should stay contacted with us just to stay abreast of what these offerings will be. Let's say something now about how we're going to run today's seminar. Let me just say if there are any technical issues that you have during the course of this, you can contact the FNIH events staff at events@fnih.org, and we have two of our staff that are helping us with the behind the scenes technical aspects of this seminar, and they can help you with that. Presentation will be roughly 50 minutes. There'll be questions after the presentations, and you can use the Zoom chat function. We will end at 90 minutes even if there's remaining questions to be addressed, and we'll address those questions offline. We are going to record this presentation, and we'll post it to the Gene Convene Virtual Institute for later viewing by you or colleagues that weren't able to join us today. Okay, so it's my pleasure now really to introduce today's speaker, Dr. Bruce Hay from Caltech. Bruce is a professor of biology in the Division of Biology and Biological Engineering at Caltech. I think of Bruce as a scientist. His interests are broad, and he creatively and successfully explores those diverse interests in his laboratory. For example, in addition to the expertise that will be reflected in his seminar today, Bruce is also pursuing projects involving the engineering of plant and animal mitochondrial genomes, as well as developing novel animal and wildlife contraceptive strategies. So very diverse interests and interesting. Bruce has been a real innovator in the field of engineered gene drive technologies. His first major contribution was his creation of the first engineered toxin-antidote type drive system that he called Medea. You may hear something about this system today, so I won't say much, but I will say that this system mimicked the behavior of a class of selfish genetic elements found widely in the genomes of plants and animals, in which the element conditions all female gametes with a toxin that will act to kill any resulting embryo that does not inherit the selfish genetic element. So mom kills her children that have a particular genotype. And of course, this is just like Medea of Greek mythology, who kills her two sons by Jason after Jason runs off with another woman. So unfortunately for those two sons, they had the wrong genotype. It was a brilliant piece of work using Drosophila as a model. And when mosquito biologists acquire some of the precise gene expression reagents needed, they'll be able to recreate Medea in them. So today, Bruce is going to talk about some of his latest innovations, which are, like Medea, highly original. So good morning, Bruce. I want you to get ready to share your screen. Thanks for joining us. We're all grateful and really eager to hear what you have to say. So thanks for getting up early this morning and doing this.
No problem.
So let me give you the ball here, and then the floor will be yours.
All right, let me get my
Go ahead.
Get my tool ready here, and let's see if I can get this on full screen. Okay, so thanks, Dave, for the generous introduction, for inviting me, and for putting this whole thing together. It's really going to be a very interesting ongoing series, and I think it's particularly important that it's all being recorded because it gives a lot of people who will only hear about it sort of secondhand an opportunity to really go back and use this all as a resource. So what I want to do today is to introduce you to a new kind of gene drive, but it has a long intellectual history in a way, and so I want to introduce you a little bit to that as well because it provides some context. And so let's let's look at what we want to do. So as Tony talked about last time, of course, one of the things we want to do is to modify a population, change its composition. The other thing that we'll hear really an amazing story about is from Andrea Crisanti about population suppression. And then later in the talk, I'm going to tell you a little bit about an idea that we've been playing with called population modification followed by suppression, in which a modified population is carries essentially a time bomb of some sort that causes it to die at some particular point in response to an environmental trigger. Now, the issue with all of these, whether it be homing or any other technology, is that we're trying to spread a trait into a population. And the challenge, of course, is how do we do that? Because Mendelian segregation normally wouldn't do that with just your regular alleles. And not only do we want to do that, but we want to do that in ways that are portable to diverse species, in which the components are genetically and evolutionarily stable over hundreds of generations and then millions of individuals. When we think about modification in particular, we want to think about the fact, and I'll come back to this later, that things break. And thus we need to be in a position to actually carry out multiple cycles of population modification. And then we also need to think about control in some way. And this can be defined in various ways, and I'll come back to that in a minute. So these are the folks in my lab who have been working on this problem. Tobin Ivy, a graduate student, Georg Oberhofer, a postdoctoral fellow, and Marlene Biller, also a postdoctoral fellow. And so I'm going to introduce you to their work, but I'm also going to introduce you to some work from some other labs that have come up with related technologies, and we'll get to that in just a second. So here's the fundamental problem again, that genes that we want to put into populations typically are going to confer on individuals a fitness cost as compared to not having it. And thus, all other things being equal, that trait is going to fall out of the population. And so the question is, what can we do as genetic engineers with some kind of drive element of some sort and a cargo that we want to move into the population that will allow us to spread the trait of interest. And the way we're going to focus on today, and this is a mechanism that nature uses widely, is to have those who carry our element of interest introduce a fitness cost onto those who do not carry it. And in this way, the relative fitness of those who carry the element becomes greater than those who lack it, allowing the former to spread itself into the population. And so the general way to think about this is that every race has a winner. You don't have to set a world record in order to win a race. You just have to be faster than the other guys. And so what these elements are doing is they're creating a context in which they have a relatively greater fitness than those that lack them. So the important concept here is relative fitness. Now, it seems like this is impossible, right? That this is some sort of genetic action at a distance. Seems like it just can't happen, like this dog walking on the wall here. But in fact, many organisms, essentially all kingdoms of life, use this strategy as a way of getting ahead, the selfish elements that they carry. And they do this in a very simple, conceptually very simple manner, though the details are often quite elegant and complicated. And the short answer is that they carry something called a toxin-antidote cassette. So if we look here at plasmids and prokaryotes, they carry a cassette that makes a toxin and an antidote that's inherited by all the cells that carry the plasmid. Those survive because the antidote is inhibiting the toxin. But whenever the plasmid is lost, the antidote now disappears and the toxin acts unopposed, killing them. And in this way, the plasmid creates a selfish advantage for itself within the host cell. Fungal spore killers do a similar thing, but in this case, they're acting in meiosis. So when a diploid undergoes meiosis, you end up with gametes that carry one of a pair of homologous chromosomes. One pair carries the TA cassette. They survive. One carries not the TA cassette. The antidote eventually disappears and they end up dying. And in this way, again, spores that carry particular chromosomes gain a benefit. And then finally, we also see this in higher animals with things like Medea, which has now been found not only in Tribolium, but multiple examples in worms as well, and ants. as well as a plant system. And these work through a basic strategy where, again, the mother provides the toxin, as Dave mentioned, to all of her offspring. And the only offspring that survive then are those that inherit by virtue of inheriting in the embryo an antidote that turns on just in time so as to be able to rescue the development of those that all carry the toxin. And what all of these do then is to create a relative fitness advantage for those who carry the element. And again, the way that this all works is this sort of magical behavior where you have action at a distance comes from the fact that these individuals are all lineally related from a parent who has the ability to provide some components in trans, meaning the toxin and the antidote, and the antidote only in cis then in those who inherit the element. So it's a very, very simple logic. Now, so what I'm going to do today is to talk to you about a new kind of selfish genetic element that uses a toxin antidote based principle, but the toxin that is moved from cell to cell in this lineage tree is not going to be a protein. It's actually going to be a loss of function allele with of an essential gene, with the idea that those who fail to inherit copies, wild type copies of the essential gene, end up dying, whereas those who inherit functional copies of the gene, in this case the green antidote, end up surviving. Now, as we look at the whole field of population-based management, gene drive, there are a lot of different kinds of organisms in addition to disease vectors that we're all interested in, and a lot of different contexts that we're interested in. And this does suggest, given the fact that these naturally occurring elements have been enormously successful in being able to spread themselves in the wild in various environments, it suggests that, well, we might want to use those as the basis for our engineered elements. And we've spent a lot of time over the years thinking about this idea, and ultimately have kind of come to the conclusion that it's quite challenging. And the problem is that each of these TA elements, it grew up in a very specific neighborhood. It grew up in the context of a particular host genes, development, and ecology. And so it works really well in one specific environment in which its mechanism of action is clearly toxic and rescuing. But the gene regulation, the translation, the titration of the levels of the toxin and antidote, those are all things that have been worked out over evolutionary time. And the ones that we see are the ones that figured it out. The ones we don't see are the ones that didn't figure it out, and they had too much toxin, not enough antidote, et cetera. And so the problem with this approach is that you really have to know a lot about how each of these components works and how that activity can be translated into the species that you are interested in. And we spent some years sort of pursuing this idea with Medea, synthetic Medea in Drosophila, and we could get it to work in Drosophila, but as a part of a larger project with Tony James over a number of years, where Omar was a postdoc in the lab, where we were trying to port this to Aedes aegypti, and in which Omar made probably hundreds of transgenics to try and get this idea to work. We just couldn't get it to work for reasons that are still unclear. And so it illustrates, in a way, the challenges with requiring a lot of knowledge about development, gene expression, the details of the organism that you're working with. And so over the years, we've become something of Luddites. We want to know as little as possible and still have our system work. And so this is what I'm going to tell you about today. The idea is a new kind of drive mechanism that we call Cleave and Rescue. It requires very little species-specific knowledge. Now, I'm going to talk about some of our published and archive papers. I'm also going to introduce you to some unpublished data, but I also want to call out some work from Jackson Champer, Philip Messer, and their colleagues, where they've developed something similar as well. They have one paper here where they do some experiments, and they also have some interesting modeling papers in which they've gone into a lot of the different ways in which the basic ideas I'm going to introduce you to can be used to make gene drive mechanisms in principle that can do modification, suppression, high threshold, et cetera, et cetera. So take a look at those, and we'll come back to the experimental paper in just a minute. So what I'm going to do today is to lead you from our data through 4 topics: population modification, the idea that everything breaks and we need to replace old content with new, the idea that we can take a self-sustaining drive and make it self-limiting and control that, and then the idea, if I have time, of conditional suppression. All right, so everything begins now with Cas9. And the very simple point, which we all know, is that you can insert Cas9 and some guide RNAs at a point in the genome. It can be basically anywhere. And they will act in trance to cleave copies of a target gene. And then the simplest thing that can happen is that cleavage will result in the breakage of the gene through non-homologous end joining. And when we think about this, breaking a gene, we always end up using multiple guide RNAs, usually 4, so that we can prevent what Tony talked about last time, this idea of resistant allele formation. And these are the alleles that are uncleavable because they've undergone a point mutation somewhere in either the PAM or the guide that recognizes the sequence homology, but that still leaves the gene functional. And so the idea is we really want to avoid this, and the best way to do that is simple redundancy. Now, everything I'm going to talk about today is with Cas9, but I do want to emphasize, and this is kind of amusing for me in a way, having worked on gene drive for a long time, that everything I've told you could have been done at some level in the mid 90s using either zinc fingers or talons. The idea is to make DNA breaks. But more importantly, going forward, it's also important to recognize that there are a lot of ways of modifying DNA that don't involve the creation of a double-strand break. And I'll just say that double-strand breaks are particularly exciting way of activating a genome to engage in various kinds of repair attempts. And DNA nicking followed by base editing or reverse transcription of a target site of a target sequence of interest into the nick. These are much more gentle ways of introducing changes and have some appeal in the sense that they may avoid some of the problems that one might encounter with double-stranded breaks. Though, as I'll tell you, we haven't actually encountered those problems. But we're always concerned about what could happen, even if we haven't observed it yet. So the basic idea again is simply that Cas9 and guide RNAs create breaks and they disrupt genes by one mechanism or another. And again, we don't really care how. A recoded version that lacks target sites for these guide RNAs is effectively resistant. And if you put these 2 simple things together, you get this thing that we call Cleave and Rescue, where we have Cas9 and guide RNAs that come along and cleave a wild type copy of an essential gene, create loss of function mutations. This cell and this organism survive because they carry a recoded version of that same essential gene under the control of its own promoter. And we take advantage of the very simple fact that for most essential genes, one copy is sufficient to confer wild type function. So you only need the one copy that you see here. You don't need for normal function both wild type copies. So let's look at then how this cleaver spreads. This is the basic element again, and then what happens is that in the germline, the element will cleave the wild type copies of the essential gene. Here we're just illustrating that they're on the same chromosome because it's easier schematically to do that. But again, the target site can be anywhere. And in our cases and Jackson and Phil's cases, we've put them all over the genome. Now, if we think of this as a maternal gamete, then the diploid cell ultimately give rise to gametes. The red is indicated by Cas9, and an important feature of the promoters that most of us use for germline expression in insects is that there's maternal carryover of the Cas9 guide RNA complexes, and those then act in the zygote to cleave the paternally provided copies of the genome, in this case resulting in a set of individuals who have had cleavage in the female germline, cleavage in the zygote, resulting in only the survival of those who carry the cleaver element because they carry this recoded rescue. These that fail to inherit the element end up dying. And so this creates this relative fitness advantage for element bearing individuals. Now, of course, the element can also come through the male germline. In the case of the promoters we typically use, there is not male carryover into the zygote, but in this particular case, it wouldn't matter because you have cleavage in the maternal germline, cleavage in the paternal germline, and this then leaves you with one zygote genotype that doesn't survive. This individual now survives because it inherits a cleaver cassette from the father. All right. So how does this behave? And what I want to say here is I want to talk about how it behaves, and I'll just say that its behavior is different than that of a HEG. So a HEG can spread very rapidly from low frequency, a cleaver type element or Medea or any other toxin-antidote system typically spreads quite slowly when it's rare. And that's because most matings are among wild type, and there are very few between wild type and the cleaver bearing individuals, and thus there's very little killing. And so when cleaver is introduced at a low frequency, it spreads really slowly until it reaches a certain threshold in terms of percent of the population. But once you start to add a bit more, it then converts the population very quickly. The bottom line is the more you add, the faster it goes. Now, again, what we're concerned about, of course, are elements that carry a fitness cost. And whenever you carry a fitness cost, in order for drive to occur, the killing of the non-carriers needs to outweigh the cost associated with whatever fitness cost cleaver or Medea or anything else carries. So the larger the fitness cost, the more you have to add in order for drive to occur at all. And again, the more you add once you cross that threshold, the faster it goes. But very importantly, of course, when you have a high fitness cost and a low introduction frequency, you just fall out of the population because you're still in this regime indicated over here on the right. All right. Now, an interesting question that we thought about when we were developing this is well, what if your target gene is not haplosufficient, meaning viable, happy in one copy? What if it's haploinsufficient or even haplolethal? And this is important because for a lot of genes, we really don't know their level of haploinsufficiency in the wild. We know that in the lab, most genes can be heterozygous and fit, but we don't know really if that's true in a wild environment. And so Tobin, looked at all of this with modeling, and what you see is that even with levels of haploinsufficiency that go all the way up to haplolethality, drive still works, and it still works very well. It comes now with a higher threshold for introduction, for success with introduction, but that can actually be an interesting thing, as illustrated here, and probably Mike Smansky will talk a lot more about this. And this is the idea of high threshold gene drive. And the idea behind high threshold gene drive is that it creates what we would call a bistable switch, where if you introduce individuals at a frequency below the threshold required for drive, the drive element falls out of the population. If you introduce above this threshold, you get drive into the population. But then very importantly, if you dilute that modified population with wild types and bring the level of transgenes below this threshold frequency, you now kick the transgene out. So this kind of idea, high threshold gene drive, is interesting in terms of control. And it's interesting to think that simply by taking a gene that had sort of a evolutionarily baked in level of haploinsufficiency, one might be able to create this kind of threshold-based drive without doing a lot of other tweaking of the drive element. All right, so that's all nice theory stuff. And this is the element. What does a real element look like? The real element looks pretty much like the schematic, and it simply consists of the nanose promoter, a germline promoter, active in both males and females, 4 guide RNAs, a recoded rescue that either comes from Drosophila virilis, a very distant Drosophila species, or Drosophila suzuki, a more closely related species, but still some distance away. And then we have 2 dominant markers, green and red, that act as our cargo. And in all of these experiments, what we're doing is we're simply taking individuals who carry this full element, and then we're crossing them to wild type and asking what happens in the next generation. And so this is the first results with the first element we built, targeting a gene called technical knockout, TKO. And really the only thing we know about this gene is that it's important for mitochondrial translation. We don't know and we don't care anything else about it other than that it's essential, small, which makes it easy to recode it. And what you can see is that a control element that just carries the rescue and a dominant marker doesn't spread, whereas the full element spreads very rapidly and basically stays at transgene and ends up very close to allele fixation forever. We targeted more recently another gene on the 3rd chromosome that's involved somehow in transcriptional regulation. You would have to read more about it than me to know what it does. Again, all we really know is that it's essential. And then we did the same thing with a 3rd gene called dribble that I'll come back to in a minute, where again, we know it's involved in ribosome assembly somehow. We don't really understand it, but we don't need to. All we need to know is that we can generate a rescue that rescues its function, and guide RNAs that cleave the wild type version. All right. Now let me. So in our hands, full elements work very well to drive into a wild type population. Now let me mention Jackson and Phil's experiments. They did something a little bit different, and there's one important wrinkle, which is that they took their cargo and guide RNAs and they actually introduced them into the target locus. So they're doing something where they're actually introducing their element into the gene that they want to disrupt. We typically stay away from that to avoid any possibility of homologous recombination, but their element is called TARE, toxin-antidote recessive embryo. They make these homozygous, and then they add into this population homozygous Cas9 that's sitting on a different chromosome, They then mix these individuals with other individuals that are wild type at the target locus and that also carry Cas9. And then they cross these, and this then is the drive experiment, to again, individuals that are wild type at the target locus and are ubiquitous for Cas9. And then what they do is they follow the fate of the tear in this universal Cas9 expressing background. And what you see, as with our experiments, is that the element spreads rapidly to transgene fixation. And so what I want you to get out of all of this from our experiments and Jackson and Phil's experiments is that is a set of rules. First, that there are a lot of different ways of doing this. We've all focused on Cas9, but it can be done with anything that can site-specifically modify DNA. Any gene can serve as a target. All of us chose different genes, so we have 4 genes, 4 successes. The rescue is simply a recoded version of the essential gene designed to be guide RNA resistant. And the only thing you need to get this going then are regulatory sequences sufficient to drive expression in the germline. And an important point here, and this contrasts with a lot of the problems that homing had in its early days, is that the promoter doesn't need to be germline specific. So long as it's in the germline, but expresses in other tissues, that's just fine, because this rescue version of the construct always rescues the survival, even if there's cleavage in somatic cells. All right, now, another point from our experiments and Jackson and Phil's experiments is that, in contrast to a lot of the early work with homing, this system is incredibly robust. So between the 2 labs, I'm sure we've looked at more than 30,000 individuals, and we've looked in genetically diverse populations from 5 continents that we got actually from Jackson and Phil. And none of us has ever seen a resistant allele. And so the bottom line is that, in contrast to homing, they just don't occur. And the reason for this, of course, that Tony alluded to last time, is that if you're targeting highly conserved sequences, and in our case, we're targeting them with multiple guide RNAs, it's simply very hard to create an allele that is resistant but still retains function. And so that's, I think, one of the real important take-home lessons so far. Now, but let's, not clap our hands too hard. Let's focus on the fact that failures will occur, and systems need to be not only robust, but they need to be resilient. They need to be able to recover from failure. And the kind of failure I'm talking about here is the fact that anything that's engineered that doesn't provide a fitness benefit to those who carry it in a biological context is going to fail. Every chain has a weakest link, and all other things being equal, stuff just falls apart in the case of biology through mutation. And this is not unique to gene drive, and I really want to emphasize this, that everything we do in the wider biological world, which is basically everything, it involves mutation and selection. And what that means is that these always come with periods of success that are inevitably followed by failure. And we see this all the way from cancer therapeutics, antibiotics, antimalarials, insecticides, and of course the vaccines that we take literally every year in some cases because the virus has evolved. So evolved resistance is inevitable most of the time. And in the context of our engineered object, we also have to think about the fact that the cargo will undergo mutations at some frequency. In addition, of course, we're going to keep learning. As we work on gene drive, many other people are going to be working on what to put in those gene drives. We're going to get better and better at that. And in addition, at various points, we may want to remove unwanted stuff that is present but no longer useful, particularly in favor of something that has a broader spectrum of interesting activities. And so the idea then is that any gene drive for population modification needs to be able to do the following. Of course, you need to be able to do the initial gene drive, but then you also need to be able to do gene drive over multiple cycles, where you replace an old element with a new element that, say, here has had a broken cargo. And I want to talk to you about how we can do this now with cleaver, taking advantage of the fact that if two cleaver elements are located at the same position in the genome, they cannot recombine with each other in meiosis. And so there's a competition between them for who ends up in viable progeny. And the basic idea that we've worked on is that we can take an old element and add to it new guide RNAs, a new cargo, and a new rescue, and put this into the old element. And in this case now, this new element should outcompete the old element because the old element lacks the new rescue. And so this is illustrated here for a female diploid cell that gives rise to these gametes. And what you see is that the cleaver N+1 survives because it carries rescue N+1, but those who fail to inherit the cleaver N+1 die because they lack the N+1 essential function. Now, we believe we can do this. We believed we could test this idea out because, in fact, all of our cleavers are located at a common position in the genome. And so we set out to build second generation cleavers. And one of them is illustrated here. And the terminology is the following, that this is cleaver dribble. And we've added a rescue to the TKO gene. And that, what we're seeing here is drive into wild type of that element. And that's what's illustrated in the red lines here. So it drives even though it carries this new cargo, which is another copy of an essential gene. Now, the real experiment is the one shown on the right, where we're taking a population that is completely fixed for TKO. It's had TKO at genotype fixation for 30 generations, and then we add cleaver DBE, rescue TKO to that. And what we see in the green lines is that the cleaver DBE rescue TKO completely takes over the population. And by virtue of taking it over, though not shown here, it kicks the cleaver DBE out of the population. So when this goes in, cleaver TKO rescue DBE, it's kicking cleaver DBE out. And I'll just skip this one, which shows another example of that, to make the point that what this tells us is that we can overwrite old content in a population with new content. And we think this is really important because it provides a way of in a very controlled manner, manipulating one point in the genome, the point at which the cleaver sits, and altering what's in that population with respect to one set of transgenes or another, in which we're always getting rid of the old element while we introduce the new element. So we're reducing genomic clutter, if you will. All right. Now let's change gears to the second to the last. And this is the idea that the cleaver is self-sustaining. It's your typical sort of gene drive mechanism that once it gets going, the idea is that it's going to keep going until it runs into some sort of physical barrier that stops it. But for a lot of contexts that people have been thinking about over the years, there's a lot of interest in the idea of having gene drive that goes for some distance, for some period of time, and then sort of peters out, where the drive is limited in some sense. And we'd like to be able to do that, but we'd also like to be able to do it in a tunable manner. in which we can make something that is almost as strong as self-sustaining cleaver, but is destined always to ultimately fail, to lose its drive potential, or something that's significantly weaker. And the way we've done this is described in this manuscript here, and I'll just lead you in one slide through the basic idea, because it's just a tad complicated. So again, with one locus cleaver, what we've been talking about, it's self-sustaining because whenever Cas9 cleaves, you create an opportunity for rescue, and that selects for the rescue-bearing chromosome. But because Cas9 is tightly linked to the rescue, Cas9 also benefits from that and is carried along with the rescue to high frequency. So essentially, Cas9 hitchhikes with the rescue when they're tightly linked, and this makes drive permanent. But you can break that relationship very easily, as illustrated in the top over here, where we've simply put Cas9 on another chromosome. Now we have a situation in which the loss of function alleles that mediate drive only are created when these two chromosomes come together in the same individual, in which Cas9 can act on the gRNAs provided here. Cas9 has no drive potential itself, and whenever it finds itself in homozygous loss of function individuals in the absence of the rescue, it's going to get kicked out of the population as a dead loss of function homozygote. So Cas9 is destined to segregate away from rescue cargo, and it's destined to be lost as the rescue cargo spreads. But we can also do something even more interesting, which is to put Cas9 on the same chromosome as the rescue cargo, but now some distance from the rescue cargo such that there's reduced recombination between the rescue cargo and Cas9. In other words, we can create different levels of linkage. And as you all know, the important thing about linkage is the closer two elements are to each other in the genome, the slower the rate at which they're going to recombine away from each other and begin segregating independently in the population. The farther away, the faster, the closer, the longer it's going to take to reach this thing called linkage disequilibrium to disappear. So let's look at how this plays out in a model, and then I'll show you a little bit of data. So here we're imagining an unlinked cleaver with Cas9 on a separate chromosome, and we're imagining transgenic release ratios of various numbers, percentages. And just focus on the solid lines. And so what you see is that when two locus cleavers introduced at low frequency, it falls out of the population very quickly, as it does at 20. But when you get up to a 30% release, you see that it actually spreads to genotype fixation for a really long time. And the question is, what mediates that? And of course, what mediates that is the fact that there's also Cas9 around. And the greater the introduction frequency, the more Cas9 there is. And it's that Cas9 that is creating the loss of function alleles that allow, indicated over here, that allow the rescue cargo, which again carries a fitness cost, to remain at this high frequency over many generations. So again, just to reiterate, Cas9 is necessary for the toxin, but the actual toxin in any sort of cleaver is in fact the loss of function alleles that are created, because it's in homozygotes for the loss of function alleles that the selection for the presence of the rescue cargo chromosome occurs. So what's remarkable here is that even though Cas9 is completely gone by generation 100, regardless of the introduction frequency, the transgenes remain near fixation. And they do this because the loss of function alleles remain near fixation. And again, it's the loss of function alleles that are always selecting against the non-cargo rescue chromosome. But what's very interesting, and again, this is kind of the key point, is that though this population from generation 150 on is fixed, essentially, at genotype fixation, it's very easy to eliminate that and to completely eliminate these cargo transgenes simply by introducing wild type, because by introducing wild type, you're introducing wild type alleles at the loss of function locus, you're introducing non-cargo chromosomes. And in particular, when you introduce the wild type alleles at the essential gene locus, in the absence of Cas9, the loss of function alleles are simply going to disappear because they're recessive lethals. And so they'll disappear with the kinetics of a recessive lethal. That removes any selection on the cargo, and the cargo rapidly disappears from the population. So interestingly, with two locus cleaver, you can keep something at fixation in the absence of a drive element, provided that you have first created a lot of loss of function alleles that can keep the rescue cargo at that fixed state. But you can very easily eliminate the individuals from the population because the true drive potential, which needs Cas9, is gone. Now, let's just make one final point about recombination. So let's now imagine a constant release ratio of 20%. So that with a 50 centimorgan freely segregating chromosomes, that's the equivalent to this blue line up here, where we have a 20% release, 50 centimorgan element. And what you see again is that the rescue gets pushed out of the population very quickly. But as you decrease the recombination distance, what you see in particular, if you focus on Cas9, is that because Cas9 now experiences linkage with the rescue for some number of generations before recombination breaks up this association, again, this is a constant introduction ratio. You see that Cas9 levels increase dramatically, though they always ultimately fall. And that's because, again, Cas9 itself has a fitness cost associated with it of 5% per allele. And thus, as recombination breaks up this association, it's lost, just as a 5% dominant allele would be lost in any population. But what's really interesting, again, is that if we go down in this simple scenario to a 10% recombination rate between these two elements, what you see is that now at a 20% transgenic release frequency, which completely fails over here, now goes to fixation for more than 600 generations. And of course, it would go even longer in the case of closer linkage. Now, but again, importantly, this drive is not permanent. It's fundamentally always limited because the source of the loss of function alleles has been eliminated with the loss of Cas9. And so if we were to add wild types at this point, the cargo rescue would disappear. Also, if there was incoming migration of wild types, that would have the same effect. It would bring in wild type alleles at the essential gene locus, and those would start the process of decay. And so the take-home message is that by introducing linkage, you can make a two-locus cleaver arbitrarily strong up the level of a single locus element. And that can allow you to get away with much lower introduction thresholds. It can allow you greater spread in space, but importantly, it's always self-limiting. And it's always self-limiting because ultimately the drive potential is lost because of the loss of Cas9. All right. So here, with that long modeling, here's just a tiny bit of data. If we make such an element and we just look at the drive of Cas9, nothing interesting happens. If we look at the rescue and guide RNAs alone, nothing interesting happens. But if we introduce the complete element, the complete two locus element into a wild type population, we see that the cargo spreads in 3 out of 4 cases to genotype fixation, and Cas9 is lost from the population and eventually just disappears again because it has a fitness cost associated with it. So from all of this then, the point I want to leave you with is that with two locus cleaver, drive can be limited in time and space. And very importantly, you can tune that level of drive, the extent of the drive, both in time and space, simply by manipulating the recombination distance between the two components of the two component system. Now, what about population suppression? That's what I want to end on here. And before I go on to our work, I want to, again, just highlight the fact that other people have been thinking about this. This is Jackson and Phil. And in one of their modeling papers, they talk about a system known as TADE, Toxin-antidote dominant embryo. And I don't want to talk about it. I'm sure they will. I just want to call your attention to it as an interesting idea for how one might be able to bring about suppression with a cleaver type system. What I want to end with is the idea of population modification followed by suppression in response to some kind of trigger. And a point I want to make is that this is not a new idea. Whitten proposed this idea in the context of using chromosome translocations as a drive mechanism way back in 1971. And so the idea has been out there a long time, but of course, for an equally long time, we haven't been able to do gene drive, and we haven't been able to engineer transgenes to give us sort of a condition-dependent lethality. And so I want to tell you about our efforts to solve both these problems. And the key to solving these problems, again, comes back to an important feature of cleaver, and that is that once it is spread to transgene fixation, every copy of the endogenous essential gene has been rendered loss of function. And so what that means is that the population, in this case, a wild population, is literally addicted to, totally dependent on the rescue transgene provided by the cleaver element. And the implication of that is that if we can somehow make the transcription, translation, stability, or function of the rescue dependent on some specific environmental or other condition, the fate of the population will also be dependent on whatever that variable is. So one of the variables that we're all familiar with, particularly in California over the last few weeks, but equally so in a sort of bimodal place like Rochester, Minnesota, is that many climates go through dramatic temperature transitions throughout the year, and plants, animals, etc., have to deal with this. And so the simple idea that we've implemented as sort of a proof of principle of this condition dependence is to imagine scenarios in which there are seasonal changes in temperature. And the idea is that what we'd like to be able to do is to introduce a drive element at a time that is permissive for drive, in which the rescue is functional. We'd like to have population replacement modification occur during that window to completion. And then as the season changes, if that's associated with a temperature transition now that makes the rescue non-functional, you should end up with population suppression. And then going on year over year, what you would be doing is introducing a few individuals. So this should result in a population bottleneck. And then the idea is to maintain the transgenic state of that population by introducing small number of individuals at the permissive temperatures when the bottleneck is just starting to open up again. So that's the basic idea. And the way that Georg and Tobin and Marlene have implemented this is very simple. And that is to take a self-splicing element known as an intene. And just as an aside, intenes are what homing endonucleases use. The HEG is in the middle, the intene's on the outside. It's what HEGs use to insert themselves into essential genes without disrupting the function of those essential genes. The idea is that the intene splices itself out, and when it does, it brings these extenes, these gene elements back together, creating a normal functioning protein. So when splicing occurs, when everything occurs well, you eliminate the intene, you get a perfectly normal protein. And so what we've done is to take advantage of temperature-sensitive versions of intenes that have been generated by Norbert Perriman, Change Tan, and others that cover a broad range of temperatures, permissive versus non-permissive, and to introduce these into one of our cleaver elements, cleaver dribble, and then see what happens. And what you're looking at here is a drive experiment where we've driven cleaver TS dribble into the population at 23 degrees. And what you see is it drives perfectly well, just like our other cleavers. And this again is all into a wild type population. The rescue alone doesn't do anything interesting as expected. Now, what we've done then, what Georg did in particular, at various points after transgene fixation, is to take adults and move them to 29 degrees. That's what these dotted blue lines are. And then we let those adults lay eggs and then look at how many progeny they produce in the next generation. And what you can see, and this again is for replicates, sorry, what you see is that some replicates produce a few progeny. Remember that a normal female produces on the average 60 eggs a day. And what we're seeing here are progeny numbers on the order of maybe 2 to 3 eggs total over the egg laying period in this case. In other cases, the number of eggs, the number of progeny that are produced by a given number of adults is less than the number of adults. So the bottom line is that adults shifted to 29 degrees basically don't produce very many progeny, 1 to 2 or less. But then very importantly, if you take those progeny that did survive, those 1 or 2 or 3 or 4 progeny, and you let them lay eggs, they're universally sterile. And what this does then is it results in a complete population crash within 1 generation. So within 1 generation of shifting things, shifting adults to 29 degrees, we can completely wipe out a population using this TS drive element. And so this shows that this kind of thing can be done. The conditional suppression is possible. And the empirical question now are what are the appropriate ranges for, say, something like Drosophila suzuki, a pest of fruit, soft fruits, for example, that lives in climates that do go through considerable seasonal temperature variation. So that's really what I wanted to say. Let me just summarize. Very simple element. Literally has 2 components, Cas9 and guide RNAs, recoded version of essential gene, that's it. You can target any essential gene you want. You can use any Cas9 RNA-guided nuclease you want. And that makes all these parts incredibly extensible in that you can use a lot of different options. So far, both Jackson, Phil, and I have us have found the system to be very robust to failure. It's also, we've shown resilience for the first time, that we can take a gene drive, spread it into a wild type population, and then overwrite that content with new. We can modify the threshold by using haploinsufficiency, and we can modify the strength and the duration of drive by manipulating recombination. And suppression can also be achieved in at least some contexts. So let me stop there, and I'd be happy to take any questions.
Thanks a lot, Bruce. Really great talk, and I'm sure you're going to be able to hear the folks' appreciation for this.
I don't hear any clapping.
Hear any clapping? That's from my studio audience.
Never mind.
Anyway, thanks a lot, and yeah. So let me just remind the attendees that if go ahead and ask your questions. If you could use the chat function, that'd be great. The question answer function we can certainly see, but the chat will be a little bit easier. I do have a question that came in. I will name this speaker since this questioner, Mike Smansky, has two question and a comment, actually. He says, you modeled two locus cleaver with a 5% cost on Cas9 or cargo or both. And with no fitness cost on the cargo, does it still disappear with time?
Great question. So the bottom line is, if cargo doesn't have a fitness cost, it does not disappear from the population. If there's migration, it diffuses out and is present at a lower average frequency. But as with any neutral allele, the bottom line is, if your allele is neutral, it's just going to float in the population based on that fact. So the answer is no. You need a fitness cost on the cargo in order for it to be lost from the population. With this, and I should say with any other of this kind of mechanism, and examples would be Daisy Drive that Kevin Asfelt is working on, Killer Rescue that Matt Scott, Fred Gould, and colleagues have been working on. All of those, if the cargo doesn't have a fitness cost, then in a self-limiting drive system, it will just float in the population.
Yeah, Mike wanted to just follow up on that too, and just aside from saying that it was a great talk, everything was good except for the slide with the Minnesota annual temperature data, and he says he hates that data.
Yeah, no, so, it's a good
and for those in the audience who don't know Mike Smansky, he is in fact in University of Minnesota, so a bit of an inside joke there. Let me have another question here. This one was actually, I'll name this colleague as well of yours, Andrew Hammond. So Drew, thanks for the question. Great talk, he says. Very exciting. What is to stop the target gene from repairing from the recoded rescue and thus create a fully resistant allele?
Right. This is a great question, and it's in a way a personal question, because I mean, we actually thought about this drive system, and Tobin started modeling it in 2014, and I kept telling people not to work on it because I was worried about exactly this thing, homologous recombination between sites distantly located in the chromosome or different chromosomes that would result in the creation of resistant alleles. And I think there's two answers to that. One is that if you look at that literature, which we now looked at a lot, what you find is that almost all of it is looking at either identical sequences that are right next to each other on the chromosome, asking, do you get ectopic recombination? Or I'm sorry, diverged sequences right next to each other on the chromosome, or identical sequences on different chromosomes. And what has not ever really been looked at, except in an evolutionary context, are sequence divergent versions of a gene on two different chromosomes, so that you have to really have homologous recombination between diverged elements. And the shorter answer is that it is possible. And it will depend on the level of homology. And so we work very hard to decrease those levels of homology below sort of empirical thresholds that have been observed for the ends that are created by cleavage of the target, that the search distance that those ends search in order to look for homologous sequences. We try and I think Jackson and Phil do the same thing, try and put enough sequence divergence in there that there are never more than like 10 base pairs of potential homology on either one side or the other. And then again, because we're typically targeting with 4 guide RNAs, we're creating actually a large region in which recombination would have to recapture all of those sequences in order to make resistant alleles. But your basic point is well taken. And the other thing to say is that when we wrote the original paper on this, we also noted that there are a lot of biological systems in which you can actually take very diverged homologs from yeast and put them, well, not yeast into human, but you can take human genes, put them into yeast, and they functionally replace a lot of yeast activities and give wild type fitness. So the point is that the sequence, the level of sequence identity can, in at least some, perhaps many cases, be hopefully reduced below a level at which we will need to worry about this. But your point is taken, and ultimately it's an empirical question that can only be determined by doing a lot of these experiments.
Good. Have another question if you got time here. We do have time for sure. So I'll also name this questioner since he's a colleague of yours, Omar. Omar says, fantastic talk, Bruce. What promoter requirements for the Cas9 guide RNAs would be necessary for building a functioning cleaver targeting a haploinsufficient gene?
Right. Yeah. So the key with anything haploinsufficient or haplolethal, I think, though one should also check in with Jackson and Phil on this. In my mind, it's that you want the cleavage to happen, the cleavage and loss of function allele to happen after the time at which the gene is used. And the problem is that when you're targeting a haplolethal, you're targeting something like a ribosomal protein, which is presumably also required in the germline. And so what I worry about personally is that if you target a haplolethal in earlier stages of germline development, then what would happen is that there would be a fitness cost to some of those germ cells, the ones in which cleavage occurred. And if there were any in which cleavage did not occur, they would have a fitness benefit, and you would get more progeny from them, and that might lead to resistant allele formation. But what I should say is that Jackson Jackson and Phil have a paper that just came out yesterday, I believe, in PNAS, which shows that they can target haplolethals in a homing-based context. But I think the homing-based context is different, because when you have homing in the germline and you carry a recoded haplolethal rescue, you're bringing in that second copy right away. so that you don't experience this fitness cost. But with cleaver, I think it would be important that the cleavage happens after the time when the gene is used, and I don't know of a promoter that does that. Yeah, because it depends, and we don't know the answer to when haplolethal products, the dependency on transcription, we don't know when that dependency on transcription ends during oogenesis or spermatogenesis.
Great. So I don't have any more questions that are coming in from our attendees at the moment. I do have a question just from my own desk here. And I guess it was a point of clarification, just to make sure that I've understood it correctly, that the cleaver system, cleaver rescue system, would always be temporary in the face of migrants coming in from outside, or would there be a threshold dependence on the migration frequency and rate?
So single locus cleaver would, well, so single locus cleaver spreads to transgene fixation. Now, and because Cas9 is always present, any wild type alleles that come in rapidly get cleaved, and the population goes back to a transgene fixation state that takes a generation. But I think that for single locus cleaver, wild type migration is not an issue in that sense, in that the population will remain at very close to transgene fixation, because there's always conversion of essential gene, wild type essential genes to cleaved loss of function that then require the rescue for survival. In the case of the two locus system, it depends. I don't have the slide here. It's in our bioarchives manuscript. But the bottom line is that yes, when you're in a two locus configuration, wild type, as with Daisy Drive, as with Killer Rescue, as with any two locus self-limiting system, when the driver, whether it's the killer, the cleaver, or the base element in a Daisy Drive, is reduced in frequency, incoming wild types will start to change the population. And this is how it's meant to be. This is the thing that actually makes these drives self-limiting, is that when they diffuse out, they lose the drive potential. But the important point is that when you have recombination or linkage, and you get that population initially to a close to transgene fixation, you actually buy yourself a lot of time at close to transgene fixation before the migration of wild types brings the level of the cargo down to something that might not be epidemiologically interesting. But again, the other critical thing to say in all of this is that all of this is empirical in the real world, right? These are simple models, and how something would behave in the real world will of course depend on what that local environment is, and how much migration there is from wild type surrounding areas. I think for any first test, and there is one, let me just add one final point that I think is interesting, because of course, we're all ultimately interested in how could you do a trial, and I think two locus cleaver is kind of interesting in this regard, in the sense that if you went to an island, let's say, and you did a two locus cleaver intervention, it's a finite population, and so what's going to happen is you're going to get rid of all the wild type alleles, and you're now going to have a population that is fixed for cleaver. But if Cas9 has a fitness cost, it's going to be lost from the population. So what you're going to end up with is literally a population that is stuck at transgene fixation basically forever, because there is no wild type migration because you chose it because it was an island. But as soon as you add wild type to that population, if the cargo has any fitness cost at all, it will just be lost from the population. So this kind of two-locus system, I think, is actually interesting to think about in the context of isolated tests. And then finally, of course, if a few individuals of the two-locus system did get to the wild type, they can't do anything because, again, drive is immediately lost as soon as Cas9 segregates in a large wild type population from the rescue locus. So it's a system that intrinsically is incredibly containable, even if individuals get out. But more importantly, on the island itself, I think you can create this interesting situation where you actually have a drive system at fixation for as long as you want, because it's the only source of essential alleles. And so you can play with that system as much as you want, but then you can kill it very quickly, simply by releasing wild types at the essential gene locus into that population.
Fantastic. Okay, thanks. We do have time for more questions, and we do have a few questions,
Okay.
more questions. And you talked about sort of what happens in the real world. This
Right.
question, I think, relates to looking down the road here. So the question is, I understand your desire not to know a lot about
Right.
the targeted gene, or perhaps the function of the rescue, at least in the process of development. But would you need or want to understand those complexities at a fine-grained level before seeking regulatory approval or an environmental release? And this is a question from one of our attendees.
Right. Yeah, and that's absolutely true. I mean, in the context of just getting something to work, which is kind of where we've been for some number of years, my excitement in conveying our excitement about the fact that we didn't need to know much really just reflects the fact that in many previous drive mechanisms that we developed, we actually needed to know a huge amount just to get it to work, whereas here we didn't. But in the real world, of course, you're very much going to want to know how that rescue transgene actually does affect the physiology of the organism. You are going to want to know, is one copy really sufficient to give wild type fitness? And does the recoding that you've done confer any other unexpected characteristics? These are, and again, these are things that you really like to be able to test in this again is why I brought up this idea of island tests, the idea that you could put something in an environment that is relevant, and you could really try and study the fitness and the physiology of the organism in that environment. But absolutely, in the real world, which is a regulatory world, and that ultimately is the most important thing, you need to know what you're doing.
Yep. Okay, got another question.
Yep.
Still have a few more minutes, which is fine, which is great. Let's see. Do you think that the climate and seasons would be predictable enough to warrant a release in the wild?
Right.
Great
Question.
There's a follow-up.
Right. Yeah.
Yeah. And what if the insects migrate? What if the insects migrate to a region that's under constant permissive conditions? And wouldn't these conditions provide a breeding ground for resistance to take hold?
That's right. They would. And so very simply, the idea of conditional suppression requires that you be working in an area in which there are enough barriers that allow you to do something interesting without having to worry too much about migration, long distance migration, long distance migrants coming in. and screwing things up. And again, the empirical question will be in contexts, particularly agricultural contexts, where there are dramatic temperature shifts throughout the season. Are there also, what are the dispersal distances of the insects, say Drosophila suzuki? What are the what are the dispersal distances? What are the migration rates, and how would that affect the system? Is it plausible that ones that migrate out to some spa-like environment that's at a constant temperature of 23 or 24 degrees would then migrate back at a permissive time to this area? These are simply empirical questions. If anyone has
there's a question from Mike. Yeah, go ahead, Bruce.
Oh, I was just going to say, if anyone
here's another one from Mike Smansky.
If anyone has positive thoughts, positive comments on contexts in which conditional suppression might work, we'd love to hear them. Okay, and then Mike.
There's another question from Mike Smansky. Have you modeled this with the cleaver system on sex chromosomes?
Yeah, we have, and it works well on sex chromosomes as well, and it will basically drive the chromosome you put it on into the population. It has not been possible in our minds thus far to use that to bring about population suppression. And so I could go on for a while about the problems that come up, but but yes, both we and Jackson have looked at sex chromosome linkage. The one difference with sex chromosome linkage targeting an autosomal gene, which Jackson and Phil show in one of their papers, and we showed with Cleaver or with Medea a long time ago, is that it drives to allele fixation rather than transgene fixation. Whereas if you're autosomal, you tend to drive towards transgene fixation rather than allele fixation. But otherwise, the drive characteristics are pretty similar. But we haven't come up with a good way of doing the kind of suppression that you're going to hear about that Andrea Crisanti is going to talk about.
Mm-hmm. And finally, is there a reference for the conditional work that you talked about?
Not yet. We're writing it up soon.
Yep. Okay. That was a question from the audience as well. So I don't see any more questions, Bruce, and I think this is a good time to sort of just wrap this up. Let me say thank you very much for your excellent talk. Really stimulating, creative, and innovative as expected. I do want to just remind the audience that we will have another webinar next week. It's scheduled at the same time and place, and our speaker will be Dr. Omar Akbari, who will talk about the advanced genetic control of human disease vectors. I think you heard his name mentioned during the course of this seminar today. I also wanted to acknowledge the FNIH event staff, both Tara and Deetria, who are beyond the backside of this, whom you can't see and interact with, who have helped to organize this and to help it run as smoothly as it has. So with that, I'm going to thank Bruce once again, and
thank you.
We'll see everybody next week. Thanks, Bruce.
Okay.